see them in cells. Here's the cartoon one more time. And you see here that this is the same stuff I just showed you. It's just over and over again, right? But once this goes, we've talked about the major translation factors here. There are other translation factors. And once this goes, other translation factors come into play and they initiate translation. This slide over here shows you one thing. I wanted to show you this slide. There's PABP1, the one that I showed you before, which is the cytoplasmic. Remember, there's a PABP2, right, which is the poly-A binding protein. Where's PABP2? Where's that found? It's in the nucleus, right? So when translation occurs, that poly-A tail is added, the B2 is added. But then after the message is processed and it's transported out of the nucleus, the PABP2 comes on. And this guy here that's made in the cytosol comes on, the P1. The P1, PABP1 comes on. So there's two different PABP1s. This guy was named first because we first found it bound to messenger RNA. Later we found it bound to hnRNA. Then we realized there were two different ones. So the one in the nucleus became number 2. Still with me? Questions? You should ask them. Okay, so here's that picture. This is something I want to show you. Here's EIF4G, right? And this is sort of interesting. Remember, 4G is involved in this initiation process. You have to have 4G to form that 4S complex that has 4E in it that initiates translation. And this just shows you how one way in which translation is regulated. And this IRE here is an iron response element. And certain proteins that respond to iron, right, that are induced by iron in cells, have these IRE elements. So this is in the messenger RNA, this IRE element. And what happens is that in the absence of iron, this EIF4G binds to that complex, say here a loop like that, and it stabilizes that complex. So the message can't be translated for 2 reasons. One is it's this hairpin loop. It's in the 5 times untranslated region of the messenger RNA for iron-induced genes, iron-induced expression. With me? So it binds like that. It has this hairpin, so ribosomes can't get through. It also recruits this 4G, which is an initiation factor, right, that normally is needed for initiation. So it's bound here. So these genes, in the absence of iron, are not translated. In the presence of iron, right, there's a protein that binds to this 4G. The iron binding protein binds to this thing here. The iron binding protein, or the protein that binds to this response element here, the iron response element, only binds in the presence of iron. Because the protein binds iron first and changes its conformation to bind to this complex. That's how it senses iron in the cell. Excuse me. So it senses iron, it binds to here, and that protein causes this 4G to release, which goes into the translation process, which initiates translation. And now these genes that are involved in proteins that are involved when iron is in a cell get turned on and expressed, right? So we all realize, maybe you don't remember, maybe no one's told you, that iron is extremely toxic in cells. Even though it's essential, right? Your hemoglobin doesn't work with iron. If there's too much iron in a cell, it's extremely toxic. Why is it toxic? Why is it so toxic? It's a redox metal, right? It screws off the redox in your cells. So free iron is always bound to proteins like transferrin, an iron binding protein in cells. They're iron binding proteins. So when there's iron gets into the cell responds by making proteins to bind to the iron so that it's not toxic to everything within the cell. And the response element senses that there's iron in the cell and then induces a translation of these proteins like transferrin that bind to iron. And once again, this is the reason that I show you this is to show you that this initiation process, right, with all of those translation factors, proffers the cell a way to very tightly regulate translation and when it occurs and how it occurs and what genes are translated. And it's done because of all stops. If it needs more, they make more RNA, which also has another minute or two, and they make more protein. But that's how they regulate how much of the protein and they make just what you need. The eukaryotic cells regulate also, and they do it by degrading messenger RNAs. And messenger RNAs have a much longer half-life. And we definitely know that messenger RNAs that have a short half-life have a shorter poly-A tail, a much shorter poly-A tail. What messages have a short half-life? Well, there's lots of them, but the two big ones that we think about are the cell cycle proteins that are made during a specific part of the cell cycle, and then their synthesis has to stop, the cyclic proteins. And it stops because the messages are degraded. The other are histones, right? Histones are only made during the S phase of the cell cycle when the cell is replicating its DNA, because then it has to make nucleosomes. So it's making all those histones. When that's done, it turns over. Histones have a very short half-life, only an hour or two. Their poly-A tails are the shortest in the business. Okay, so the poly-A tail is related to the half-life of the RNA. And what happens is this process here, there's an enzyme that's called DAN that is also part of this complex, this initiation complex. And DAN is a deadenylase. Right? DAN is a deadenylating nuclease. It's a DAN, deadenylating nuclease. And what it does is that every time this initiation process occurs, and the poly-A tail is near this initiation site like there, and DAN is part of that complex, DAN takes a bite on those poly-A's there, just like the background figure there, right? He removes 20 or 30 or so residues. Remember, as this cartoon is fairly accurate, each of these PAB binds about 3 or 4 adenosine residues. So if you take off, 20 to 30 residues, every time translation is there, DAN takes those off. You shorten that poly-A tail by 20 or 30. You shorten this poly-A binding protein by 4 or 5 units. And so every time the messenger RNA is translated, it gets shorter and shorter and shorter. And there's two reasons. One is for regulation, right? It determines the half-life of the RNA. When those are gone, the RNA is not usable. So that's a regulatory function. The other is that remember that this process, this translation process that occurs inside of the solid cell with ribosomes that are kind of crude machines is rather a vigorous process. And during the process, these messages can be destroyed. So what half-life also does is it ensures that the messages that are being translated are fairly fresh, that they haven't been damaged or cut off or whatever. So not only does it regulate, a way to regulate which proteins are made by degrading the RNA, but it also ensures that damaged RNAs aren't making useless proteins. Okay. So once this tail gets short enough, then the poly-A binding proteins can't get there. Once it's shortened to about 20 or 30 or so residues, poly-A binding protein can no longer bind A residues and stabilize this cap binding process, and the RNA is turned over. Now, there are other ways in which RNA, the half-lives of RNA can be regulated. And one thing, if you remember, these guys have a 5' cap. Poly-A tail protects them from the 5' exonuclease and the 3' exonuclease. But if you generate a piece of RNA that doesn't have a 5' cap or tail, then it's very quickly degenerating. What happens here is it shows that as the poly-A tail shortens, what happens is it sort of allows endonucleases. For some reason, that poly-A tail, right, and wrapping it around to form that polysome protects the inside or the inside. So this part of the RNA, right, is involved in this process here. So as long as this tail is 20 or 30 nucleotides, it's like this, and somehow this formation protects RNA in this region here also. We're not sure how. Maybe because it's attached to ribosomes. Right? You stop initiation, there's no ribosomes there. But anyway, once that tail shortens, then an endonuclease is able to cut in the middle. Now, I'm not sure why your book does this. It does it in a couple figures, right? Where if you look at this guy and you cut it in the middle, then it flips it. This guy is over here, and this end is over there. Right? There's a poly-A tail, there's a cap. I don't know why they do that. But anyway, once you do that, it takes this part here, and it exposes the 5' exonuclease. Maybe that's why they do it. Here's the 5' end. There's not cap, 5' exonuclease chews in. Here's the 3' stabilize this 5' cap region. There's a decapping enzyme that can remove the cap, which activates this nuclease, but also you can activate an endonuclease that cleaves it and does that. So either way, poly-A tail is gone and the RNA is degraded. This is just one of the problems. There are actually some mRNAs that actually have a sequence or cleavage, endonucleolytic cleavage sequence in their 3' untranslated. Remember, the dark blue is what's translated. This is the 3' end of the messenger RNA, and it's not untranslated. It can be 600 nucleotides, 500 nucleotides, whatever. It can be long. And a lot of them within that sequence have this endonucleolytic cleavage site. And the half-life of the RNA is determined by whether there's an